Today we will talk about the infections caused by staph and streptococci. And we will see, when we talk about staphylococcus, there are many strains, right? So we need to differentiate them, whether it is Staphylococcus aureus, whether it is Staphylococcus epidermidis. We will name only those microbes which are involved or belong to lab objectives, okay? Similarly, for the streptococci, we will see how we identify the streptococci and the species or what we call the strains within the streptococci, right? Like you have a streptococci group A, group B, group C, group G. At the same time, you have the streptococci, there are no grouping. Okay, some streptococci like viridans, you have mutants, you have mites. So how to differentiate them? How to identify them? In general, if you say a slide, you can say this is a streptococci, but you need a further identification to confirm whether it is the streptococci pneumoniae, whether it is streptococci pyogenes, whether it is streptococci, so on, so on, so on. Okay, so today we will talk on those microbes, and they cause many infections. But here we are concerned as a cardiopathogens. Okay, that affects your heart, either the membranes of the heart or walls of the heart. Okay, so these are the objectives we have to cover up. You see here, Staphylococcus. This is a huge family, micrococci family. It involves Staphylococcus aureus and Staphylococcus epidermidis. So you have to differentiate how we can differentiate them. Staphylococcus aureus and epidermidis from each other. Despite they belong to the same family. So this means there are differences, maybe in their enzyme production, maybe in their fermentation of sugar, or maybe type of a hemolysis, okay, that we see on the blood agar. Similarly, viridans, streptococci, you have viridans, strains, many mites, and we have mutagens and so on that cause infection. How you differentiate this from streptococci pneumoniae? How you differentiate it from the streptococci pyogenes? These three belong to the same family, Streptococcus family, Staphylococcus micrococci family. So we have two families, but within the family there are the microbes, and you have to differentiate them in the lab as per the tests needed. Finally, Borrelia. I have put one slide for this microbe also. So we will go one by one, right? So let us talk first about the Staphylococcus aureus and epidermidis. How to differentiate the two and how to isolate the two. Is there any difference or no? We will see. As I said, in terms of, because if you see under the microscope, you will find Staphylococcus grape-like structure. So at that moment, you can say it is a Staphylococcus aureus, but you cannot say, is this a aureus, is it the epidermidis, is this another or what? So you need further examination. So let's go for Staph aureus and Staph epidermidis. When you see under the microscope, this microorganism, it is cocci-shaped sphericals. Under the microscope, it's a gram-positive microorganism. Both Staphylococcus aureus and epidermidis, when you go for the enzyme test like the catalase, it is positive. So what's the catalase test? Okay, tell me.
Conversion, it's to
breakdown of hydrogen peroxide
Correct. Breakdown of hydrogen peroxide
to which?
to water.
Yes. Once the microbe infects the body inside, the same time the neutrophils come to attack that particular microbe, right? But at the same moment, the neutrophils, they release many things. One of the things is hydrogen peroxide. What it's called? Oxygen reactive species, right? Or you call them ROS, and nitrogen reactive species. These species mean these are the unstable. They are called as free radicals. Once this is released, this can cause immediate death of the microorganism, destruction of the cell. So H2O2 is one of the free radical. This is produced by neutrophil in order to destroy the bacteria. But if microorganism is having the enzyme catalase, the microbe, when H2O2 approach the bacteria, immediately H2O2 job is to destroy the cell, okay? But immediately, if microbe is having the catalase enzyme, immediately the catalase is released by the microbe. It affects on the H2O2 produced by the body to destroy the organism, but it converts into oxygen and water. So bacteria is safe. This is a protection to protect themselves, bacteria, having the nature provided them the enzyme catalase. So we see Staphylococcus and Staph aureus both are catalase positive. So those microorganisms cannot be affected by highly reactive oxygen species like H2O2. There are also the nitric oxide, you have heard the name. Also the highly unstable the molecule, it can also attack nitrogen species, but at that time the catalase enzyme also is effective to neutralize the free radicals. The short, remember, job of catalase enzyme is to protection for the microorganism. It neutralizes the free radicals. So we have seen here Staph aureus and Staphylococcus epidermidis, both microbes are catalase positive. Okay? Now next, coagulase test. But here is a difference. Staph aureus is coagulase positive, epidermidis is coagulase negative. Okay? Here is the difference. So what is the coagulase?
Coagulation.
Breakdown of fibrin clot.
Breakdown of
Fibrinogen into fibrin makes the clots.
Clot formation.
This is again the protective mechanism for the bacteria. Once the microbe is inside in your body, it causes the infection. The bacteria immediately, if bacteria is having the coagulase enzyme, it produces the coagulase surroundings, and it causes the fibrinogen to convert into form the clot around itself,
Yes.
and it protects the microorganism from the phagocytosis. So it's a kind of a protection. And here we see microscopic observation is the same, cocci shape, spherical, catalase enzyme is the same for both of the microorganisms, but as far as the coagulase test is concerned, it is present in Staphylococcus aureus and not in the other Staph. Always remember, catalase positive, coagulase positive is only Staphylococcus aureus, only microbe in this class. Okay, here Staphylococcus aureus. The God has given both the two things, the catalase enzyme and coagulase enzyme. Fine. Up to here it is clear. And here you see the clot formation, because it's a rabid plasma, okay? And you put in the plasma, you put this microorganism Staph aureus, and within a couple of hours you see the clot formation. This slide test is very quick test. You can see the clots, but tube test is having the very specific and very accurate results in the tube test. So next morning when you see there is a clot formation here, you say it is Staph aureus. And in another, when there is no clot formation, you call it Staph epidermidis.
Doctor, what the other picture?
This is the slide test, the same thing. This is the tube test. You put in a tube in the serum and you incubate it. For 3-4 hours, you find the result, but it's a quick reaction, okay, on the slide. Done?
Victor, if there is a clot aureus,
Staph aureus, Staphylococcus aureus positive test, coagulase. Is written. Other staphylococci like Streptococcus epidermidis negative test. So no coagulase formation, okay? Next.
Yes.
This is the clumping factor agglutination reaction. Again, very quick test, you can find it here for Staph aureus. And the reaction is here, it's shown. It's also in the previous reaction, okay?
It's not specific, doctor?
No, this is here.
No.
It's just, because you know, what's the agglutination test?
Antigen antibody reaction.
Antigen and antibody reaction. The coagulase test is particular for for enzyme, okay, for enzyme test. But this is for antigen and antibody. Quickly
Just
explain.
indicate that there is a bacteria, that there is a staph. Rather,
When you do the previous two tests,
no, this one.
yeah, then you can do one more. This is one more test for identification of Staph aureus. For example, you suspect from the coagulase test, okay? You found the catalase positive, coagulase positive. If you do this clumping factor test for Staph aureus, you will find this test. Again positive, it gives the more confirmation that microbe is Staphylococcus aureus. Now here, that was the test that we do on enzymes. This is a test we see on the culture media on the blood agar. On the blood agar, there are two things. The Staph aureus, it produces the golden colonies, golden yellow colonies, while epidermidis produce white colonies. Staph aureus is beta hemolytic and epidermidis non-hemolytic. Okay, can you tell me why there are the golden colonies in the Staph aureus? Okay, anybody?
Fermentation of
Breakdown of
fermentation
Just remember, the Staphylococcus aureus on a blood agar, when Staph aureus produces the golden yellow colony, this means at that moment the microorganism releases the pigment. Releases that? This golden color is because of the pigment. When Staph aureus is grown on blood agar, it produces golden yellow colonies. Golden yellow colony is because of the pigment released by the microorganism, by the Staph aureus in the medium. While as the here Staphylococcus epidermidis, it produces the white colonies. On blood agar, it produces the hemolysis, okay? Beta hemolysis, complete destruction of Staph aureus, but here in epidermidis, do not. So another technique, another test, laboratory culture, that gives you the proof, this microbe is Staph aureus, this microbe is epidermidis, okay? Blood agar test. On blood agar white colonies, here, these are the golden yellow colonies. Next slide. And this test is very important. This comes in most of the exams also. Whatever.
mouse. Clicking down.
Mouse.
Doctor, about the culture, how much time does it need?
Correct. Usually the microbes have a culture from 24 to 72 hours. But usually for these pathogenic strains, most of the strains, within 24 hours you get the results. You have a culture plate. This is the blood agar. When you do the catalase test is positive, coagulase test is positive, the clumping factor you found in Staph aureus, you stab, you make like streaking and put it in incubation for a night. Next morning when you come, you will find, or after 48 hours, you will see the golden yellow colony. Then you will say, oh yes, this is a Staph aureus. And you will see the beta hemolysis, breakdown of the RBCs on the culture or the blood agar. This is the proof microorganism beta hemolytic as well as pigment produce. It produces golden yellow pigment. Now come to here. Mannitol salt agar. This is very important media for differentiation between the Staphylococcus aureus. You see this media. This media contains, by its name, it contains two important things. By the name of the media, MSA, mannitol, salt agar. Two things are there. Mannitol, what is mannitol?
Sugar.
Sugar. Salt is sodium chloride. This medium contains mannitol and the salt. It contains high sodium chloride. Usually, microorganisms cannot survive the sodium chloride concentration of 5%. Usually, microorganism pathogenic strains, if you increase the salt concentration in the media, the microbe will not grow. But Staphylococcus aureus has a capability to grow at a higher salt concentration, and it has a capability to ferment mannitol. So you see here, when Staphylococcus is grown on the mannitol MSA agar, it produces pink to yellow colonies. Now, why yellow colonies?
Pigment.
No, just remember, pigment is produced only in the blood agar. But here when the color is changed, it is because of change in the pH. When it ferments the mannitol, when mannitol is fermented, the bacteria produces,
Lactic acid.
bacteria can produce acid, bacteria can produce neutral compound. When bacteria produce the acidic compound, lactic acid, any kind of acid, as what's the name? Dr. Bhaji said, this acid, when it's produced in the surrounding media, there is a pH, actual pH of the media, usually 7.2, this pH goes down. Usually the pathogenic strains, they grow at pH 2, okay? No, not 2, sorry, pH 7.2. So when bacteria is grown in a particular media, it ferments the mannitol. When it uses the mannitol for the energy, of course there is an end product, okay? Every reaction has an end product. So in this fermentation process, the end product is the acid. When acid is produced, this pH, high pH, it goes to the lower values, masalan 4.2. pH is decreased. When pH is decreased, this means acidic nature. And this media contain a dye, neutral red. That neutral red makes a reaction with the acid and causes the change in color. Change in color is yellow.
So
But
it was decrease in the pH.
yes. And why decrease in the pH? Because of fermentation of mannitol. Because of fermentation of a, you can say fermentation of a sugar. Fermentation of sugar, pH is decreased. pH is decreased, this means acid is more produced. More acid is produced means it reacts with the dye present in the medium. That the dye present in the medium as an indicator, it reacts with the acid and causes a change in color. Okay? And you see on the mannitol, yellow color because of change in pH. On the blood agar, you see the golden colonies because of the pigmentation. Correct. These are two different things. Okay. So when we talk about the epidermis, no mannitol fermentation. This microorganism cannot ferment the mannitol at all.
So normally it is pink.
It is pink to yellow. Yes.
No, normally before putting the strawberries.
The culture is pink in nature.
The culture medium,
Yeah.
mannitol salt agar, it look like the pink color. It look like this color. You know, this color is now changed. It become now yellow because the medium of the color, it's written here from pink to yellow. This means the medium looks like pink, but actual this the medium, the final color of the growth of the microbe, it is yellow. Okay, we will not focus on the initial color. We will focus on the final color. Okay, because on blood agar, if you see the McConkey, if you see the blood agar, it look like same by this color. Okay, but we have to look the end after the growth of the microbe. What is the final reaction? Okay. So finally, second one, epidermis. There is no mannitol fermentation. There is no change in color. It remains as a pink color. Okay, it remains as a pink color. This is the mannitol. I think we have them. This salt agar in our laboratory, microbiology lab.
You talk about the Staph aureus when it can it survive the low pH?
Survival here means when the microbe grows. First microbe grows at pH 7.2. Okay, remember pH grows at 7.2. When microbe utilizes the mannitol, it produces the acid. That acid causes low pH in the medium. Okay, low pH in the medium, and it causes the change in color. The microbes cannot survive within the optimum pH range. If pH goes more down, microbe can be killed. Only some microbes have a capability to survive an acidic pH. Okay, but microbes, Staph aureus, cannot grow in the low pH medium. And here we are not growing the microbe in a low pH. We are growing the microbe in a pH of 7.2. It utilizes the mannitol, produces the acid, and that causes a change in the pH. Okay, that causes a change in the pH. This means once medium mannitol is finished, there is a higher pH in the medium, of a low pH in the medium, more acid. Automatically within day 1 or 2, microbe will die. Okay? But important is here that if you go to the microbiology laboratory, you can see the medium. Two things are mentioned there. First is the components of media, and second is the pH, because pH is very important for microorganisms for their survival. So mannitol test confirms. Mannitol salt agar confirms that Staph aureus can utilize mannitol and epidermis cannot. This gives proof the Staph aureus can survive higher salt concentration as well as fermentation of the mannitol. Okay, this is the chart. It will explain you final what we have read till now. Staph aureus, Staph epidermis, catalase. I said both are positive. Coagulase, positive, epidermis negative. Colonies and blood agar, gold and yellow, white. Hemolysis, beta hemolytic for Staph aureus, no hemolysis for epidermidis. Mannitol fermentation, positive for Staph aureus and negative for. Okay?
Yes.
Questions? No. Huh? Next. Now let's talk about the streptococci. When you see about this, the family of those microorganisms, they are very notorious, and you know most of the things are related to the streptococci. We have seen the streptococcus on the blood agar. This is just the overview. Okay, we will go one by one of these microorganisms. Alpha hemolytic, beta hemolytic, gamma hemolytic, based on culture on the blood agar. When you culture the streptococci on the blood agar, you will find three types of things: either alpha hemolytic, either beta hemolytic, either gamma hemolytic. Alpha hemolysis is the green in color. Beta hemolysis is a complete. Gamma hemolysis no. But here, as far as our objectives are concerned, we will talk about the pneumonia, okay? Strep viridans and pyogenes. This is our concern, okay? So I put it in this figure as whole, but our focus is alpha hemolysis pneumonia and viridans, and beta hemolysis by pyogenes, group A and group B. Fine. Why alpha hemolysis it's a partial hemolysis? Why it is of green color? Because I put it as a green here. Any idea? You have read about the alpha hemolysis. You have read about the beta hemolysis. Alpha you call it a partial hemolysis. Beta hemolysis you call complete. What does this mean? Partial green, and what does complete hemolysis mean? Complete destruction of RBCs means beta hemolysis, and partial hemolysis means
Alpha.
partial destruction of what? RBC is not completely destroyed. RBC is not. Its cell wall of the, because we have cell membrane inside. It is cell membrane is attached to the RBC. It is called as partial. But why it is green? You see the green color. You have seen the alpha hemolysis is a green in color.
Yeah.
Yes, because you have lab the first year, you have labs in the second year. Alpha hemolysis looks green. You know why? Alpha hemolysis looks green because when microbe makes the partial destruction of the RBCs, what will happen? This is your RBC, and the membrane is not removed. Cell membrane is not removed, or it remains intact. At that time, partial breakdown of the cell membrane, it reacts with the oxygen present inside, okay? And it forms methemoglobin. Methemoglobin gives its green color. This is the reason partial hemolysis is a green in color. Beta hemolysis is the complete destruction of the RBCs, and you do not see any kind of the color there. Yes. No.
Partial breakdowns in the cell membrane, then react to the oxygen.
Partial hemolysis means partial breakdown of the RBC is broken down, but cell membrane of RBC remain attached to components of the cell. Okay, because RBC is a cell, right?
Yeah.
There, of course, there is an outer membrane. When microbe makes the beta hemolysis, complete destruction. When partial, this means some fragments of the membrane of cell membrane attack and it makes reaction with the oxygen. And it forms methemoglobin, a compound, and it gives its green color.
Parts of the cell membrane reacts with the oxygen. No? Parts of the cell membrane reacts with the oxygen inside.
Actually the whole cell, but the partial part of the cell membrane remains there because hemoglobin, it contains iron, right?
Yeah.
And right, it makes a compound there because why it reacts? These are the fragments of the cell, fragments of the RBC that reacts. When cell is completely utilized, there is nothing to react. So this means when I make from small circles, if you see this is the RBC, right? This is RBC and these are the fragments of RBCs. Now this is called partial hemolysis. RBC is destroyed but not completely. These remaining parts of the RBC, this outer membrane, it reacts with the oxygen, it forms methemoglobin and causes the partial green in color. Okay? Fine? So we will talk about first the pyogenes, group A, streptococcus. When you see the streptococcus pyogen, you find it round over like this is the form of change, this is the gram-positive bacteria, okay? Gram-positive. On the blood agar, when you see small pinpoint, transparent, called is beta hemolysis. Beta hemolysis, complete digestion of our red blood cell contents surrounding the colony. Group identification, how you identify it? You put in first is the blood agar. Streptococcus pyogenes or blood agar beta hemolysis, okay? One test. Another is the antibiotic sensitivity test. You put the bacitracin disc, okay? Bacitracin disc. You have seen antibiotic discs. These are like this small.
Yes, yes.
And you put the antibiotic disc on Muller-Hinton agar, and you see it is sensitive. Which one? Pyogenes.
Resistance against.
Pyogen is a group A streptococcus, okay? Based on the antigen classification, lensified. I think you had this in the lecture. When you grow this microorganism on the blood agar, pyogenes, you see the beta hemolysis, complete destruction of RBCs, and second, you will see it is bacitracin sensitive. This means this antibiotic is effective against this microorganism. Here you see. Something is going in your mind. Do you have some question? No? Okay. Here you see, this is the beta hemolysis. Many pictures. All pictures correspond to the same meaning that pyogenes, microorganisms, bacteria, streptococcus, it causes the beta hemolysis, complete destruction. You have seen here like this, okay? The colony is, that's it. There was one more test. It is the P/R test. I did not put it because it is based on the enzyme test. Production of the enzyme, it takes much time. So, but this is the important. We will talk about only the blood agar.
The right one is the those two.
A,
Can you just identify each picture?
sorry.
Can you just identify each picture, each culture?
This is all pyogenes on blood agar.
Yeah.
Single micro in various forms. Okay.
No.
Here you have seen beta hemolysis. Okay. You see at the D picture. Just to call you complete hemolysis. You see here.
Mm.
And this is the bacitracin. Antibiotic. Sensitive. Effective. This means this antibiotic is effective against the streptococcus pyogenes. Now this point is important. Other beta hemolytic streptococcus usually are bacitracin resistant. Okay? Pyogenes shows the beta hemolysis on a blood agar. This means complete information. Okay, second, when you put a bacitracin of 0.4 units of bacitracin, this it shows the sensitive, effective against the pyogenes. But when you talk about the other microorganisms that cause the hemolysis, you usually found they are resistant to bacitracin. So this antibiotic test, sensitivity test, is important in the differentiation of beta hemolytic streptococci. Those streptococci that cause the beta hemolysis, The pyogenes is bacillin sensitive, others are resistant. Others are done. Now, important test for pyogenes. Streptococcus pyogenes.